Technology issue about cloning. Cloning is a process of producing genetically identical organisms that already occur in nature. Cloning is a sexual reproduction method. In nature, microorganisms such as bacteria and plants reproduce asexually or by cloning method. Cloning in biotechnology is intentional cloning of an organism or molecular cloning of DNA fragments. In biotechnology, scientists mainly use three types of cloning, known as reproductive cloning, therapeutic cloning, DNA cloning. Reproductive cloning, cloning which are used for producing the exact copy of an existing organism are called as reproductive cloning. Reproductive cloning is also performed using a technique known as somatic cell nuclear transfer. During this procedure, nucleus is extracted from a somatic cell. Somatic cell is any cell from the body other than the germ cell. Then the nucleus of the egg is also removed. The extracted nucleus is then transferred into a nucleated egg. Egg starts to divide after receiving the external stimuli. Egg will provide and grow and finally will develop into an embryo. This embryo is then implanted into a gestational surrogate mother. Reproductive cloning has some issues like clones which are produced have shorter lifespan when compared with the parent species. Also emphasizes the risk of losing the genetic diversity of the nature. DNA cloning is a molecular biology technique which is used for the creation of exact copies or clones of a particular gene or DNA. DNA cloning is used in genetic engineering to create microorganisms, plants, or animals with desirable features, also used in DNA fingerprinting and also in gene sequencing. Steps of DNA cloning. DNA cloning is used for many purposes, and the basic steps are: first, cut open the plasmid and paste in the gene. This process relies on restriction enzymes which cut DNA and DNA which joins DNA. Second, insert the plasmid into bacteria. Use antibiotic selection to identify the bacteria that took up the plasmid. Third, grow up lots of plasmid carrying bacteria and use them as factories to make the protein. Harvest the protein from the bacteria and purify it. Cutting and pasting DNA. One common method is based on restriction enzymes and DNA ligase. A restriction enzymes is a DNA cutting enzyme that recognizes specific sites in DNA. Many restrictions enzymes makes staggered cuts at or near their recognition sites, producing ends with single-stranded overhang. The DNA ligase is a DNA joining enzyme. If two pieces of DNA have matching ends, ligase can link them to form a single unbroken molecule of DNA. In DNA cloning, restriction enzymes and DNA ligase are used to insert genes and other pieces of DNA into plasmids. Our goal in cloning is to insert a target gene into a plasmid. The plasmid, which has a single cut site, the target gene fragment which has a cut site near each end. Then, to combine the fragment with DNA ligase, which links them to make a recombinant plasmid containing the gene. The bacterial transformation and selection. Plasmid and other DNA can be introduced into a bacteria, such as the harmless in labs, in a process called transformation. After transformation, bacteria are selected on antibiotic plates. Bacteria with a plasmid are antibiotic resistant, and each one will form a colony. Colonies with the right plasmid can be grown to make large cultures of identical bacteria, which are used to produce plasmid or make protein. Protein production. Protein production is the biotechnological process of producing a specific protein. The bacteria serve as miniature factories, cutting out large amounts of protein. Once the protein has been produced, the bacterial cells can be split open to release it. The target protein must be purified or separated from the other contents of the cells by biochemical techniques. The purified protein can be used for experiments or, in the case of insulin, administered to use of DNA cloning. DNA molecules built through cloning techniques are used for many purposes in molecular biology. Biopharmaceuticals. DNA cloning can be used to make human proteins with biochemical application or biological macromolecule or cellular component such as blood product used as a pharmaceutical. Second, gene therapy. In some genetic disorders, patients lack the functional form of a particular gene. Gene therapy attempts to provide a normal copy of the gene to cells of a patient's body. Third, gene analysis. In basic research labs, biologists often use DNA during to build artificial versions of genes that help them understand how normal genes in an organism function. Therapeutic cloning. Biotechnology cloning used for the purpose of medical treatment are known as therapeutic cloning. Therapeutic cloning is mainly used to grow organs which can then be used to replace the non-functional organs of patients. Therapeutic cloning, like reproductive cloning, is also done by using somatic cell nuclear transfer techniques. Therapeutic cloning plays a major role in medical treatment. Doctors, with the help of therapeutic cloning techniques, can grow replacement organs for damaged part of that patients. Therefore, it reduces the usage of immunosuppressant drugs and also the risk of organs rejection by the body.